Okay, so see, did you get your model protein to model in Adobe Beta?
Yeah, I got it and I downloaded the data.
You downloaded the data.
Yeah.
Fantastic. Then okay, then
But
okay. So
should
today
I
we're
share the
gonna,
link with you?
yeah,
Should I share the link with you?
yeah, you can share it. You can share it. Just simply share the four models with me. You can simply mail the model. I'm going to take a look at it. And so let's today analyze that how to learn about this PyMol and what can we do through this PyMol. Okay.
Okay.
So first of all, as you open a particular thing in PyMol, it's going to show in this background. This is going to be the general background, and we can alter and change this background everything. So
Yeah.
first of all, the first thing is that you see the lower most corner we have a sequence called S, right? That gives
Yeah.
you the sequence of the whole protein. Okay, and as you select the residues there in the sequence panel, as you select the residues, you're going to see that those residues are actually marked in the protein, right? Okay,
Yeah.
but this in this case you're going to find that suppose since I have marked this area, it has marked some other area in the black space. Why is that? Because in this particular condition, this conformation is only for a cartoon model. This is a cartoon model.
Yeah.
Okay, so when if I change this to a ball and stick model, it is going to change, and you can view the amino acids individually. And just this just looks a pretty. This is just a pretty face of the whole protein, because if I make it
Yeah.
into a stick format, then it's become really cumbersome for a whole or a long or a big protein. Okay.
Okay.
So sometimes it is really hard to picturize this whole PyMol in the black background. So what can we do? Just in the display, click on display, and then in the upper topmost panel you can see the display bar, right?
Yeah.
Then just scroll down. You can going to see an option called background. Just click on the white, and it is going to turn white. Okay, and if you for zooming in and out, simply holding the left mouse button, so the right mouse button, and simply scrolling in and out, scrolling in, moving the mouse up is going to make it unzoom, and moving the mouse close to your body is going to make the whole protein zoom. Okay, you can zoom in and like that. And if you select the left mouse button, and you can rotate the protein to get the perfect view of your whole protein or the particular residue or particular motif much clearly. Okay.
Yeah.
Okay, are you able to do it?
Yeah.
Okay, so now there is a scroll. If you are using mouse for PyMol, it's advisable to use mouse. Okay, always use mouse, because the touchpad in your laptop is just not it's not does not work good with the PyMol. Now when you in the mouse you have a scroll bar, and in the scroll
Yeah.
bar if you simply scroll it down, you're going to see that the protein is going to fade away.
Yeah.
Okay, and when you scroll it back up, it's going to showing the whole proper structure. And why it is important? Because sometimes we're going to see why it is important in the later period. Okay, so
Okay.
these are the things that like this you can navigate the whole protein, you can move it around, you can simply place it around, and one more thing, if you simply press down the middle mouse button or the scroll wheel, you can simply translate the whole protein to
Okay.
some part of the screen, as I'm doing it. Okay, now you see there are several panels or boxes called ASHLC on the right
Yeah.
side. Okay, and there are two options. There's one is called all, and beneath all you are going to have your model. Okay,
Okay.
the model you select is going to have that particular model. Now
Okay.
anything that belongs to all, if you click any button that belongs to all panel, then it's going to change everything that is on your screen.
Okay.
Why it is important? Because sometimes you are going to use a complex of molecule, right? When we dock your particular protein with a DNA, you are going to you are going to see that you are going to have two entity in your screen, a DNA and a protein.
Yeah.
And then whatever we do with all, it is going to affect both the protein and DNA together.
Okay.
Okay, and that's why if you need to see modify only the protein molecule, then you can select only the protein molecule, because at that time you're going to see that protein and DNA as a separate entity here. Okay, so now what does this button mean? Now first button A is the action button. Okay, just click on it, you're going to see it's the action button.
Okay.
These are the things that we can do. We're going to look at action a little bit later. The next thing is show. S is for show. Okay, now in show means how does your molecule look like. Okay, so first if I click on lines, if you select on the protein and click on lines, then what will happen? You can see the amino acid in lines format.
Okay.
Right, now you see all your amino acids are there. You can see the side chains, you can visualize the side chains. Earlier the side chains were missing to make it as a pleasant view, and it just shows you the backbone of your whole protein molecule. But as you select the line, it is going to show up the whole what is it, amino acid side chains also. Now you see, if you select the A option of the particular alanine in the sequence or any amino acid in the sequence, it is going to make sense, right? It is going to select all the residues or all the atoms of your that particular amino acid residue.
Okay.
Okay, yeah. Now like the line, there is also option called stick. If you click on the stick, see, these are the much more thick lines. Stick means a ball and stick model, much more thicker lines. And why it is required? Because sometimes now if you are having your large protein, right? So if you just click on stick, it is going to be cumbersome. You are not going to you are unable to understand what amino acid is which amino acid, right? It really becomes hectic for you to navigate around this whole protein while keeping every amino acid in a stick format, right? So that's one of the bigger issue. That's why we don't use stick much. But yes, sometimes we use stick. And why do we use stick? We use stick because sometimes when you are going to dock these two proteins, you are going to find that two proteins are making interaction, and generally they make interaction via this amino acid side chains. So what are we going to do? We need stick, because when we need to find out the point of contact or the residues that are interacting with one another, then we can modify each and only those residues into a stick format, and we can see how much far they are apart, what type of interactions are they creating, is it a hydrogen bond and all, and like for that purpose we need that stick one. Okay, but apart from that, and that stick is also required for publication purpose, to publish this data, to get a whole picture, nice picture of your molecule, and that's all. Otherwise making stick, it makes really cumbersome. And in the molecule also, just do it, just click on H, just beside S, there is an option called H, right? Click on H, and then the H button is for hide. It will hide anything that is going to show you. So you have selected stick, now you have just click on H and select stick, it is going to hide the stick, right? Okay, now one thing is that if you see that in your molecule there are diverse set of colors, right? There is blue, red, yellow. Okay, what are these? Generally the blue means it's a nitrogen.
Okay.
Anything blue, the atoms which are marked in blue color, they are all nitrogen atoms. Okay, anything blue, they are nitrogen. Now what is red? The reds are the oxygens. So you are going to find that it is going to have the C double bond O, right? The C and every C, H are going to be in a white present. All of the H are going to be in white, and if you are unable to see that, please make it stick, so it's much better visual. You can visualize them much better. So make it stick, and you are going to see that the blues are for nitrogen, the H stands for hydrogen, sorry, the white stands for hydrogen, the greens are for carbon, okay, and the red are for oxygen. Now there are going to be two kind of reds. Just see my screen once. See, this is one kind of red.
It's the C double bond O and the
this one is the C double bond O. And what is this one?
Um,
This is the OH. OH. Okay, so because C double bond, it has two double bonds, so that's why it is mentioned as two. You can see two bonds, right?
Yeah.
So it's C double bond O, and we knew the other part of carboxyl is OH, and this is the OH part. And since OH doesn't have any double bond, it's a single bond entity, so it feels like this OH. Now in case of OH, you are not going to find the H option here, but you can add it. But generally this means OH. And apart from that, we are going to have something like this yellow. Can you see some yellow colored residues?
Yeah.
What is this?
Is it the disulfide
Yeah,
bond?
right. These are the cysteines. Actually, not disulfide bond. It shows you the SH bond, the cysteine, the sulfur. Okay, it marks the sulfur. It marks the sulfur. But yes, sometimes whichever protein have disulfide bond, you are going to find an exact replica of this particular yellow bond just opposite to that particular bond, because they are making a disulfide bond. Okay, so that's about the stick one and all. Now apart from that, you can see another factor called ribbon. Okay, now let me hide the stick, let me hide the lines. Okay, now then another option that you have below in the show button is the ribbon. Okay, now ribbon means, what is ribbon? Do one thing, click on H and click on everything.
Okay.
Click on H and click on everything. Okay, so it is going to erase everything on your screen.
Yeah.
Now click on show and click on ribbon. So this is the ribbon structure that you are seeing.
Yeah.
Okay, this is just the ribbon structure. It's going to show you how the protein just looks like in the ribbon format. But this format is not that useful, because we are not going to use or take a look into that particular factor here. But sometimes people use this ribbon structure to for publication purpose. But that too I don't feel good, because I simply use the cartoon model. Now if you simply click on the cartoon, click on S and click on cartoon, you are going to see the cartoon model. So this is the cartoon model that you have. Okay, and this is much better, because in the cartoon model you can visualize each and every other domain as a distinct domain. You can identify the alpha helical region, you can identify the beta sheet region, you can identify the random coils and turns and all.
Yeah.
So that's why cartoon is much more visually pleasing, and generally people we also use, and people also use these cartoon figures for publication. And apart from cartoon, you are going to require mostly you are going to require the stick format or the stick model, just to make the point of interaction and for that purpose. Now in the show option, we are also going to have some other options, like there is option called sphere. Okay, if you click on it, it's going to make you a ball of spheres. Okay, so why it is important? But can you understand anything from the sphere?
No.
No, you are unable to see anything for sphere, because the sphere is not meant for everything. Just do one thing, select only a particular residue from your sequence panel, or simply click anywhere on your screen. It is going to select a particular residue. You done?
Yeah.
Now, as you select a residue, see there is an option going to become as SELE, ACLE, just below your protein name.
Yeah.
Now that means this particular button is for only the selected atoms. Okay. Now just to make aware of your for your publication and to make other people for your presentation purpose, now if you click on the S button beside the select or the selected atom, and now click on sphere. Now see, it is going to mark the particular residue as sphere.
Yeah.
Right? So only these particular residues are important. This is really important, because sometimes, as you know, in protein structures and all, sometimes you're going to find some unique residue that is responsible either for the agglutinations or the aggregations, or to make a very important connection, or to mark the exact active site. Suppose it's an amino acid present at the active site and a really important amino acid. So you can mark that individual amino acid with this particular sphere option. That is going to highlight that particular residue. But if you select the whole molecule with sphere, it is not going to make any sense to anyone. Okay.
Yeah.
Now you can also change. There is an option. Now since this is, you can see that it's actually highlighting the whole residue. Now there is an option to select and deselect this whole residue and make only a particular select a particular atom, not the whole residue, but to select a particular atom. Okay, I'm going to see how to do that. Now just check in the top, not bottom right corner, in the bottom right corner, just where we have the sequence panel, just above it you are going to have an option called selecting residues. Right? So that means I have selected the residue option here. Now I need to deselect this residue. If I deselect the residue, then it is going to select the particular molecule. Now simply click on that, right click, let's just left click on that particular residue, and it is going to change. Right? Now if you change it to atoms, if you simply change it to atom, now click on anywhere on your screen. Now see, it is not going to select a residue, it is going to select only the atom where you are clicking. Okay, so now I have selected a particular atom, and now I am going to again click on that particular show option beside select and click on sphere. Now see, only that particular atom has been marked. So this is much more visually pleasing, and this is much more required because it is the atoms that make the connections, the interactions, right? So to highlight the atoms in the publication, we are going to mark them. For example, suppose just let me see, select this. Okay, so I am selecting this particular residue. Just wait, let me show you how to do that. I am selecting this particular residue, and I am selecting this particular residue. Okay, now suppose in a way these two are making a connection. Okay, suppose these two, this is a OH group, right? You can see the O and then a H. This is a OH group and this is a hydrogen group. So suppose this is making a hydrogen bond. Okay, so what I'm going to do, I'm going to change this residue, selecting residue, I'm going to make it selecting atoms. I'm going to select this particular O and I'm going to select this particular H. Okay, now I'm going to go to the select option and I'm going to make both of them sphere, and see, I have made a sphere. So that means I'm selecting that they are making a hydrogen bond. Okay, so this kind of interactions make sense, and these are only done actually with this sphere option, just to highlight and make a particular atom sphere, so that people, because usually it's really difficult for people to understand what atoms are you selecting. So if you make them sphere, it is people are going to understand what atom are you selecting, and they can see that it's an oxygen and that's an hydrogen because of the color coded. Okay, okay, so I'm again going to make it now you can, apart from atom, you can change it to anything. Suppose there is an option called chains. Now it is only going to select a chain, like this chain A, chain B, but since this protein is only made of chain A, it is going to select this whole protein. If I keep it, then there are segments. Segment means a particular set of segment. For example, if you select this segment, so select, it has selected this part of segment. If there are several domains in a protein, right, and if you change that to a particular segment, then it is going to select only the particular domain that where you are clicking. Apart from that, you also have objects and then molecules and then C alphas. Okay, generally we need atoms and residues, and apart from the atom and residues, we don't require anything else. Okay, wait, so let me hide everything first. It's yes. Yes. Okay, so now again in the show option, we are going to have other option called, now after this we are done with spheres, right? Next we have is the mesh. What is the mesh? Mesh simply tells you how your molecule looks like in a 3D format. In a 3D format. For example, if you see that only this is your protein. Wait, okay, this is your protein. Are you looking at my screen? Okay, so this is your protein. So generally the proteins are globular, right? Now sometimes it might be it might occur to you that when I'm interacting two proteins together, does it have any grooves there? You can see this is the groove, right? This is a groove. But this particular thing is not going to be a groove, because this
Okay.
is much deeper than that. Because this particular protein, it has some level of interactions. That means apart from this whole surface, it is also going to make some interaction beyond a certain distance from its surface.
Mhm.
And how long is that surface going to be? That's what we are going to know about from mesh. Like
Okay.
till this point the protein can make interaction, though you can see that the protein has here, but but its side chain protrudes through these regions, right? These are the side chain protruding regions, and that means that till the domain the protein is, you can see these domains, and this is the actual protein,
Okay.
and these are the actual groove. You are finding that this is a groove, right? This is
Yeah.
particular groove, but apart from that, you can see these are the other kind of major grooves. You can see, right?
Okay.
These are the other
Yeah.
kind of major grooves that the other proteins are going to exploit. And this is required because just to show you give you an idea of the surface of the whole protein, and also the secondary structure of the protein.
Okay.
And since this is a mesh, this is just like a net, so you can see what's beneath it. And apart from this whole protruding regions, you can see what's underneath it, and you can see the secondary structures, if it is beta sheet or protein, alpha helix and whatnot. And even in the mesh, there are options. You can see the color codings. There is red, there is blue, right?
Yeah.
And red and blue means these red areas are the areas where the oxygen is present.
Okay.
Okay, whereas the blue are the areas where the nitrogen is present.
Okay.
Now this simply means that suppose there is a protein with another a nitrogen group. Now can you say in what areas can that nitrogen interact to? In this area,
Sorry.
suppose you have another protein, okay, and in that
Yeah.
protein you have a nitrogen.
Okay.
Now to what domain that nitrogen is going to interact to?
Probably the oxygen region.
Probably the
Oxygen region.
right, only probably on the red regions, because
Red.
the
Yeah.
red regions are red, but it also tells you that the red regions are the electronegative areas.
Okay.
The red areas are the areas where the protein has electronegative charges, whereas the green ones are the areas where the protein have electropositive charges. So therefore, any type of interaction you are going to see, you are going to see that a red region is generally going to interact with a blue region.
Good. Uh-huh.
So that, because that's a electropositive electronegative interaction.
Okay.
Okay.
The green areas are electropositive or the blue areas electropositive?
The blue areas are electropositive.
Okay, so the green areas are
Green areas are the carbon areas.
okay, carbon areas.
Yeah, they have carbon over there, and generally that particular area does not have any charge. Okay, that green areas, they generally don't have any charge, so they can make any kind of interactions. But only the red regions are going to make some electro make interaction with the blue areas, electropositive
Yeah.
electronegative interactions. And the green areas are the uncharged areas. Okay, because generally in protein molecules the carbon has filled up valency,
Yeah.
all its valency are filled up, so generally it doesn't have any charge or any kind of valency left to make any interactions. Okay, so apart from mesh, there is also option called sphere. Sorry, surface. If you click on it, what's the difference? See, this is the surface of the protein. This is how your protein originally looks in your body.
Okay.
Okay, but the drawback of this is that you are not going to find you can't see the secondary structure beneath it,
Yeah.
right? You can't see the secondary structure, but this is how exactly the protein looks like in your body. Now in this particular case, in the mesh, you can see the secondary structure, you can get an idea about the surface of the protein, but see, the surface of the protein is much better visualized in this surface area,
Yeah.
right? Now let me hide the mesh. Okay, see, this is much more clearer, and these are the areas, these are the red areas are the electronegative areas, and these blue are the electropositive areas. And see, the grooves are much better visualized, right?
Yeah.
And generally protein interaction occurs through these grooves. you are going to see another protruding part of protein interacting with is fitting into this groove. And generally that will be the electronegative or the electropositive part, because this particular groove has electronegative charges. See, this whole groove has electronegative charge.
Yeah.
So that means any kind of any protruding part, if suppose a protruding part like this has the electropositive elements, right? So another protein, if that particular thing has electropositive charges like this, it is going to simply fit into this particular groove and make a very excellent connection. Okay.
Yeah.
And this is how the protein looks like in your body. When you are going to purify a protein or crystallize a protein, this is what the protein will looks like. And you will be unable to see what's beneath it. You are not going to get the cartoon theme and all. And it will depend upon the experiment you perform to get the idea what's beneath it and all. Okay. And this is the structure that people generally use when we make some protein-protein interaction, just to give an idea that yes, every as I've told you that how do protein make contact? Each groove fits into another grooves, right?
Yeah.
And this is particularly visualized, that can be well visualized in this surface mode only. And that's why generally when we make protein-protein interaction shown to people, we make this kind of interaction. We make this surface area. Just to give you an example, just let me show you. Because okay, just let me show you. So this is the interaction of three protein molecules. Okay, HDAC1, SMART1, GATA2. You can see that all the grooves are fitting into each other, right? All the grooves are fitting into each other, and that is best visualized in this surface area, and that's why we need this surface. Okay, and see, we can identify this. These are the spheres that I have made, because to mention that the lysine 102 buried inside. This particular thing had something to do with lysine. The lysine 102 was really important, and so I've made, because people will be unable to see the lysine residue, right? And that's why I have made it a sphere. That is the role of these spheres and all. When should we do it? Why should we do it? And you are going to get the idea when we work with this, right? And you can see other modes. See, this is the other mode. I have made it and why is stick required? See, this stick, I have made this a whole surface. Okay, but beneath the surface, you can see I have made this. I have made only these three residues as a stick format. And in the stick mode, it is really understandable that they are making a interaction. Okay, this kind of interactions are really required, because two other proteins. This particular residue belongs to SMART1 protein, and this belongs to the GATA2 protein. And only by if this is in a surface mode, we can't know for sure which amino acids are interacting with which amino acids. But I have made this surface a little faded away, and I have used this stick model to map that yes, they are really interacting. Okay, and that is the requirement of this whole things. That why do we need this kind of all different kind of modes for the visualization purpose. Okay. Now apart from that, this surface has another very important role. Can you say it? What's the important role this surface has?
The surface, I'm not sure.
Okay. See, as I mentioned you that the protein originally, the protein looks like this surface. It looks like this, right? So sometimes whenever you model a protein, suppose you have modeled a protein, okay, as like you have modeled your particular FOXP1 protein, right? And generally the protein has a globular state, and when you simply have your different models, if you make them surface, you're going to find the overall general globular structure, but sometimes this particular globular structure is missing from the modeled protein, and you can see a long arm extending like this, a long arm extending, a long arm extending. And when you make it a surface, you're going to see that that particular area has a long thing, and then you have a small region here. So just by looking at it, you can know that it's not going to be the actual model. Okay, it's not going to be actual model. Because when you click on surface, you're going to get a brief idea about how your protein looks, and generally the proteins are globular. Generally, not always, but generally the proteins are globular, and so it does not have any abrupt structure. Okay, it generally has a circular round shapes. Generally, as most of circular round shape structure, and it does not have an extended arm like the chromosomes and all. Okay,
Yeah.
that's also one thing we need to check for surface, where it gives you an idea about the particular protein.
Okay.
And okay, so now let me hide. So the hide means everything, right? The hide has everything that you can do, everything, show and hide. Okay, and
Yeah.
apart from that, you have this L shaped, and that's a level option. I'm going to show it later on. And then you have the C option. The C option is for coloring your protein.
Just.
For example, just you have the idea about the coloring by element. So if you click on, there's various kind of colors. That means in this kind of color, what you have, I have selected this one. So this one means that the carbon will be green color, hydrogen will be white color or the gray color, nitrogen will be red, blue, oxygen is red, and sulfur is yellow. Okay, apart from that, I have a different color shape. That means the carbon will be pink, and rest of them will be same. Okay, so suppose I am clicking it, and by clicking it, I am going to alter the structure and the color. Why? Because it has marked all the carbon as pink, and generally, since this one is not showing you the sticks, if you click on sticks, see the color mode has changed, right? So this is the way you can color, because sometimes if you are going to have more than one protein, then we can't keep all the protein with same color. It is going to confuse the readers, right? It is also going to confuse you. So generally, when you have more than one protein, you have more than one chains also, A, B, C, then you can color them accordingly, simply to identify and distinctly visualize each of them. Okay. Now, apart from coloring by this kind of elements, you are also going to have a color by chain, and that's only when you have multiple chain, ABC chain. Then you can simply do go color by chain, and it's going to change the color of each chain differently. Okay, for example, since this is only a single chain, it has marked everything as with blue. And if I select this color by chain, one thing is going to get lost is the coloring by element. And see, everything is now green. Everything is now green. The carbon, the nitrogen, the oxygen, the sulfur, everything is green. Next, you have the coloring by the secondary structure, and that means it is going to color all the secondary structure differently. The helix will be red, the sheet will be yellow, the loops will be green. You can do, since we have a protein, I think you have multiple domains with both alpha sheet, beta sheet, and your alpha helix. And do it, you are going to find different type of colors. Just simply, since I don't have any other kind of options here, you can do it with your, okay. Done?
Yeah.
Okay. Now, other way you can also color it according to the color. You can, there are various color mentioned here: red, green, blue, yellow. I mean, even in red, you can see there are number of different color gradient.
Yeah.
Right? Salmon, red, deep red, blue red. I didn't know this kind of red also occurs. Chocolate brown, everything kind. And then you have these greens and all. But my favorite is actually cyan.
Cyan.
Yeah, it's my favorite color. You are going to find this particular cyan everywhere in my papers. I really love this cyan. And and also write this wheat in the tint color. I like this wheat structure, wheat color. It's a really soothing. It's just my favorite. It has nothing to do with its personal choice actually. Okay, and you can use this and simply use this to color your whole protein, because sometimes when you're going to design a picture, then obviously we need to have this kind of, you need to give a picture or a color that is going to be that people can visualize, and it's going to be eye pleasing, right? I'm not going to make it a very deep red, but yeah, sometimes you require this deep red colors. You require this deep red colors because we all need this. So this is about the whole coloring and all. Okay, done.
Yeah.
Okay. If you want time, you can use this on your. You can do this on your own time, right? I'm just showing.
Yeah.
Okay. Now after that, there's an option that I've missed out. There's the action button. And what is this action? Okay. Now let me to visualize the A button. Let me first open multiple structure. Okay. Now see, I have opened multiple structure here, right?
Yeah.
Now, just to show you the idea about this whole protein, just it sticks. Okay. Now see, now all these proteins, they aligned with one another?
No.
They are not aligned with one another, and it's really difficult to predict a particular structure and to compare the structure of two proteins if they are not aligned together.
Yeah.
So first of all, we need to align it, right? How can we do so? Simply click on the action button on the second model. So, you have different kind of models available. Just load all the models to your screen. Simply click on file, click on open, and load all that. Select all the models apart from the model that you have already selected, and do it. And it's going to open all the models together in your screen.
Yeah, I think it opened. Yeah.
Okay. So are they all aligned?
No, they are not.
No, they are not. So it's really difficult to compare all the models together, right? So now just click on the action button of the second molecule, second model, and then come and you are going to find the option called align. Okay. Then go to two molecule. Under align, you are going to find the option called two molecule, and under two molecule, select the model 1.
Okay.
And see, the second molecule is going to be aligned with the first model. Now again, like that, simply click on the third model and make it align to the first model. And like that, again click on the align option to the fourth model and make it align to the first model.
Okay.
Do it. Done?
Yeah.
Okay. Now see, as you align them, you are going to see some yellow lines, right?
Yeah.
Are you seeing these yellow lines? So what are these yellow lines?
They interact in the different models.
Yes, the yellow lines show how much that particular model is differing, or how much it has been translated. How much that particular atom or residue has been translated. Okay, but we don't require those. So simply see, just beneath your models, you are going to find options called ALN full model 2, ALN full model 3, 4, 5, like ALN full.
Yeah.
Right, simply remove those ALN and it's going to remove all those yellow lines.
Okay.
Okay. Now this is your aligned structure. Now if you look at it and you can really compare the two structures together. For example, I have hide. Now simply how to hide and select the models. When you have all the 4 models, simply clicking any model, deselecting any model is going to hide that particular model. Deselecting another model is going to hide that particular model. And again clicking it is going to bring it on the screen. Okay. So I'm going to, since I'm only aligning, comparing 2 models together, I am deselecting the model 3 and 4. Okay. Deselect model 3 and 4. Simply click on the model name.
Yeah.
Just click on the model name and they will be deselected. Done?
Yeah.
Okay. So now see, now I can compare these 2 molecules. So now by comparing these 2 molecules, I know that these domains are actually perfect. Now these domains is actually perfect with aligning with one another. And overall the loop is also kind of similar, right? The loop is almost kind of similar, this particular loop. But the region that it is varying is the region starting from here, because it is not making an alpha, it is making a loop till here and then it's having an L-shaped structure, whereas this is a perfect U-shaped structure. Okay. So this is why it is varying. So now if I select another model and a model 3 and I am selecting model 3, now see this also has the same domain like this particular helix is probably the helix, because everyone is forming this party part here. And next we have is another helix here with a very short loop, then another loop, and then another helix here. But if you take a look that it also has an L-shaped structure, right? So let me align it with the model 2 model, the second model. Now if I align it with second model, see, these are lining up. The loop is also similar, but the dissimilar thing is that the orientation of this other alpha helix. The first alpha helix has a different orientation, while this particular pink alpha helix has a different orientation, right? The orientation differs between these two models. And if I simply add the model 4, now see, the model 4 looks like this model 5, first model. I'm going to compare them and see, these two looks more or less similar. Now you can see that in this particular domain, this particular alpha helix has shown to match with all the 4 models, right? Yeah. That means this particular alpha helix has, this is the unique structure of this particular model. This is the alpha helix structure of this whole model. But this structure here, this has a particular different shape, and this is where the problem lies. And in this case also, it's not a very great difference, it's a very little difference, and that means both the models are really absolutely similar to one another. Okay, so like this, you can simply compare these two models together once you upload all the models, and this is and we require, and we can align it by using the action button. Okay, now other command of action button is to, suppose, now deselect all the models, just keep on selected only a single model. Okay, do it. Okay, now simply come, and there is an option called find. See, just above align, option called find. And on the option find, you're going to have a option called polar contacts. Okay, now select polar contacts and select within selection. Now it is going to show you all the polar hydrogen bonds with yellow marks. See, can you see the model hydrogen bonds, yellow dash lines? These are the hydrogen bonds that a molecule is making. This is the hydrogen bond that your molecule is making, but you are going to find these hydrogen bonds are unfinished, right? Why it is? Because if you simply go on show and click on lines, you are going to see now it is not going to look like an unfinished model. See, if you click on the option called stick or lines, it is going to mark the amino acids side chain, and now you can see the hydrogen bonds that is actually present there, right? So this hydrogen bond is actually present between this residue and this residue. And what are these residue? See, these are the aspartic acid and the lysine. This hydrogen bond is between aspartic acid and lysine. And similarly, this hydrogen bond, these two amino acids are also together, and this and that is the alanine and valine. So this alanine and valine is also making a hydrogen bond. It's a same chain, but this is simply locking these two molecules at these two ends together. This particular hydrogen bond is locking these two ends together. So this is another point. This is another option called to find the hydrogen bonds. And sometimes what you have, suppose you have two complex. Okay, you have two proteins. But if you do it on a large protein, you are going to find multiple yellow lines, right? You are going to find multiple yellow lines, and that's really difficult to understand what is projecting with which, and because to make a sense of this particular line also, you have to, or make sense of this hydrogen bond, you have to select the stick option, right? And selecting the stick option, have you selected your molecule with stick option? It's going to show you a number of yellow bonds, and that is going to make it much more problematic, right? But still, you have the option to zoom in and all, and do that to identify the unique residues that is making participating in this hydrogen bonds. Okay, like this, simply zoom in on everything and to find out the exact locations. So this is one of the options. This is the leucine, and this particular thing is the tyrosine. Okay, Okay, now in the A action, there's another option in the A, that is the simply
I
click.
have a question.
Yeah, same.
So whenever there are whenever we start when we get four models of the same protein, do we
I can't hear you. Can you be a little louder?
yeah, so when we are trying to predict a protein structure, so we have four models. Do we
Yes.
always use all the four models? Like, do we put them in Primal and then put them on top of each other and see if they are aligned or not, or do we just use the model that's most appropriate?
No, generally when you have four models, it's better to look at all the four models together, align them up, and see if anyone is mismatching. You can see the mismatches, and usually what happens is you are going to find the particular model that suits you, and you are only going to select that particular molecule only. But
Uh-huh.
sometimes the four models are indistinguishable from one another, and if
Yeah.
you align them together, you are going to find that exactly they all the models are similar. And if all the
Uh-huh.
models are similar, the work becomes much easy for you, and you can select
Yeah.
any one of the molecule
Uh-huh.
and go from there. Okay.
Because the protein that I got, all the four models were totally different. Only I think model two and three.
Just do one
Uh,
thing. Can you share your screen with me? It will be much easier for me to understand. Let me stop presenting. Just share your screen and tell me, then it will be much easier for me to understand what you are saying.
you see my screen now?
Yes, I can see it.
Can you see it now?
Yeah, yeah, yeah. Sure.
Yeah. So these are just two of the models that I had.
Okay.
Because the other two models they seem quite off. They were elongated, so we said if
Okay,
they
okay.
look around,
Yeah,
they're
fine.
probably not right protein structure. So these this is model two and three.
Yes, fantastic. Can you align them together?
Yeah, so we used action of align.
Yes. Now simply click on that ALN domain. Uh, no, click on that ALN domain model. That
Yeah.
no, just beside it. Deselect it. Yes.
Okay.
Yeah. Done. Okay.
Mm.
Okay. So this is your two models aligned, but there are still some differences. But you can't exactly say. Now simply deselect one model. Yeah.
Yeah, I think this is model two and this is model three. Oh,
Okay.
actually, yeah.
As per as
Really
I
like
see, I think the earlier model was much better.
for the earlier one.
Earlier one.
This one.
I think the earlier one was much better.
Yeah.
I can show
This
you
one,
for sure. I can say you for sure if you simply select. Just send me the models to me. I will
sure, I'll
take a
send
look
it.
at myself and then tell you. Okay.
Sure.
Okay. And apart from that, there is another option too. Simply, there is an option called Molprobity that we can use to simply identify and understand what better this thing is and how to visualize it much better. And
Okay.
we're going to talk about that particular server software in the next class. Okay. And
Sure.
that will also give you a much better idea to how to select and look like.
Uh-huh.
Okay. Okay, but as per as my option is taking between these two models, I think this is the much better one.
This is the better. Okay.
Yeah, yeah. Okay,
Mm-hmm.
but I need to look at it. I'm not 100% sure, but okay,
Sure.
send me and I will go for it. Okay,
Sure.
okay. Okay, so this is the option to simply find out the polar contacts. And now one drawback is that if you have any water molecules, sometime when you're going to do some molecular simulations and all, and then you are going to have a lot of water molecules, right? We're going to have,
Yeah.
I'm going to show you that we're going to have some water molecules because we need to perform the simulation in a water-based, in a water environment, because our body is full of water,
What?
right? So we can't perform the simulation in vacuum, because there's a lot of other help that also the water gives you. And once we start with the um, drug designing part, then we are going to learn about why water is important, what water is important, and all. Okay. And when water is present, you are going to find numerous and multiple hydrogen bonds, and that's going to simply mess you up.
Okay.
Okay, so and how to get rid of it? Simply click on H. See in the hide, just click on hide option, and see there's an option called water.
Yeah.
Okay, simply select that water, and whatever water molecule will be there, that will be gone.
Okay.
And after that, you can use this your molecule to find out the polar contacts of your individual molecule. Okay, now there's another option is to simply click on action button and go to generate, just beneath align. And in
Okay.
the generate, you have an option called vacuum electrostatics. Okay,
Okay.
and under vacuum electrostatics, you have an option called protein contact potential. Simply click it. Done.
Yeah.
Now this protein contact potential gives you the actual idea of how your protein is going to behave originally.
Yeah.
Earlier I was telling you that this portion is red, so it is going to bind to a nitrogen and all, right? But this is the actual electrostatics of your protein.
Okay.
That means this whole portion, the deeper the blue, the more is the electropositivity. You can see, see this is the 84.464. This is the how much is that your electropositivity is around. So these are the much blue region, deep blue. That means these are the point contacts that is going to make excellent interaction with the electronegative domains. So in case of my protein, but see the red region is really less, right?
Yeah.
So that means more of and moreover you can see that this electropositive domain is shifted towards only a particular domain, only to a particular side, right? All the other domains, they have a light blue color. That means they are electropositive, of course, but not like this particular domain here. And all the other protein, you can see this is the red one and this is the red one. So that means it is only this two region through which it can bind to another electropositive domain of a protein. But mostly this particular protein is positively charged amino acid, and therefore it is only going to interact with a protein which has a net negative charge. Okay, and this is the actual electro what should I say, electropositivity or electronegativity of your protein. And this is the actual thing where the protein is going to make a contact.
Okay.
Okay, so just having a nitrogen or oxygen group is going to do nothing. This is the overall vacuum electrostatic potential that you have, and this is really required because when you find some protein-protein interaction, if you simply design the vacuum electrostatic potential of both the proteins, we are going to find that this particular domain is only inserted to a domain where it is rich in oxygen.
Yeah.
Okay, and that is going to make your point much more provable that yes, this domain is actually interacting with this particular domain. But if even after this electrostatic, we are going to find sometimes that yes, that particular domain is also not that kind of electronegative on the other protein. So that means that particular domain might not interact in your system also. But if you find the accurate result that matches with your hypothesis, it's going to be much better, right? So that's another option that you can do with this action button. This particular action button here to generate the vacuum electrostatics, and it's really required to study the protein-protein interaction or protein DNA interaction, because you know that the DNA is negatively charged, right? So generally, if this particular protein, in my case, if this particular protein is a transcription factor, then what do you think, with which surface it is going to bind to DNA?
Sorry, what was that?
Suppose this particular protein is a transcription factor, okay?
Yeah.
Just can you say with which domain it is going to bind to the DNA?
So DNA is negative, so it's gonna
Yeah.
bind blue.
Yes, so it is going to bind to this region. So that also tells you how the protein is going to behave or how the protein is going to interact with one another, right?
Yeah.
And that's why we need this vacuum electrostatic potential. So you can simply, just by taking a look at it, you can understand that with which amino acids are going to be responsible to bind to to your DNA, okay, and all. And apart from that, there is another option called let me see. Yeah, there is another option called option called hydrogen. See, sometimes we are going to have, as I mentioned you, in the X-ray crystallographic structure and all, you are going to have some PDBs that is not going to have any hydrogen in it, on it, because why do you know why is it so? Because hydrogens are really permeable to the X-rays. The X-rays can simply penetrate the hydrogen. Hydrogen is not going to deflect any X-ray. For example, if my oxygen is going to deflect an X-ray because of its rich electrons, right? It has a very, in its nucleus it has a lot of electrons. It is going to deflect the electrons, deflect the X-rays. But hydrogen fails to do so because it does not have one, right?
Yeah.
And it is not strong enough to deflect the X-ray, and that's why generally the hydrogens are missing in an X-ray crystallographic structure. We are not going to find any hydrogen because it's really difficult to place hydrogen. So there, but for protein docking studies and all, and to see the hydrogen potentials, where is the hydrogen, and without hydrogen, can I predict the hydrogen bonds? No. So that's why I need to sometimes add the hydrogens, and you can do so by simply click on action button, and there's an option called hydrogen, okay? And see, if you simply click on add, it is going to add all the hydrogens on your proteins. And you can visualize it if you simply remove, just hide everything, hide all these things together, just select all your protein, and for example, in my case, let me be selected by element, okay. So this white are the hydrogens. So in my case, I have the hydrogens added, but and during molecular dynamics simulation also, each and every atom is taken into account, okay. So it's going to perform all the calculations that it is performing for carbon, nitrogen, oxygen. Is going to perform the same kind of calculation for hydrogen also, right? So usually what happens, it makes the whole calculation far-fetched, and it takes it becomes computation intensive to calculate every actions and properties of this hydrogen. So usually before molecular dynamics simulation, we remove the hydrogen to reduce the computation time. And how to do so? Simply click on this action. There is an option called hydrogen. Then under hydrogen, you have the option called remove. See, I have selected removed, and all the hydrogens are removed from the system.
Yeah.
And if I now save this particular molecule, this particular molecule will be devoid of hydrogens. And if I run this particular simulation, it is going to take a lot of less time, because the computer is not calculating the hydrogens, the forces behind hydrogens, okay. But sometimes that's not unique, because if your whole protein contact is based on hydrogen bonds, then the requirement and the presence of hydrogen is essential, right? And for that, sometimes the crystallographic structure, they are missing the hydrogen. So what we can do, click on action, click on hydrogen, and then simply click on add. It's going to add all the hydrogens. And similarly, like all the hydrogens, you can also add only the polar hydrogens, and you can remove the non-polar hydrogens.
Okay.
And why the non-polar hydrogens are removed? Because usually the non-polar hydrogens don't make any contact with the water. They are not exposed outside, right? And since the non-polar hydrogens are not exposed outside, it's 90% of the non-polar hydrogens are not going to make any active contacts with another protein or with the water molecules outside your proteins. Therefore, to keep those, if you need to make your computation power lessen, but not that long, you simply need to remove the non-polar hydrogen, but keep the polar hydrogen, because those are the hydrogen that is going to make the contacts, right? So that's why we read this hydrogen also is an important thing. And since I've told you about water, right? Hide the waters. Yes, I have hide the water. You can also hide the hydrogen. See, under the hide option also you have hydrogens. All are non-polar. How what to hide and what not to hide. Then third option you have is the label. The label is option just to simply label everything. If you simply click on residues, it is going to simply label all the residues together. See, it is only going to residues. Alanine 6, lysine 10 and all. And these are the labels it's going to make your label. And apart from that, you can also label the chains. So this is, it has mentioned that this is chain A. This is chain A, chain A, chain A, chain A, and everything like that. And it's also, if you select this atom name, it is going to mark all the atoms. H13, H12, Cg, Ct, Cb. Okay, and these are generally not required because we don't mark the atoms, but sometimes it is really important, because if you are going to make a protein contact, suppose there is another protein here, so you know that this CD1 is making an interaction. So if you simply label it, you know for sure that this is the CD1 that is making the contact. So it's good for writing papers and all. Okay, that's why. And simply like this, it's also, I have told you about B factor. How much flexible your molecule is, or that particular domain is, and see, the higher the B value, that means higher it fluctuates. Now take a look at it. See the molecules inside this helix. What is the value that they usually have? 1.08, 1.42, 0.87, like very low value, right? But see the B values of these amino acids in the loops. 5.82, 7.06. That means that's extremely high. That means they fluctuate a lot. But since these are structured, they can't move a lot. Okay, so they have a very constrained domain. So with this B factor also, you are going to, now just by taking a look at the structure, you're going to know which amino acid is going to have a high B factor and which is going to have a not. But see, in this particular loop also, this particular protein in the loop also, see this one, these amino acids are also in the loop, right? But see, the B factor is 1.25, 1.46, 1.46. So that means even though this is in a loop, they are not fluctuating a lot. They are not fluctuating a lot. But see this particular thing here, but this one is in a loop, but it is having a high B factor, right? 7.06, 5.31, but not this one. So that means even within the loop, there are some amino acids that is going to maintain their positions. Okay, but now it also depends upon the type of model that you have selected. And now if you select a second model, see in the case of second model, it is going to see that you are going to find that B factor of this region is really low, right? And since it is so low, because in this case you can see this is as a B factor of 1.89, 1.95 is low enough, and that's why this model, this second model has actually confined that to a semi-helix region because of the low B factor. And it assumes that since in a loop, amino acid can't have a very low factor, but since these two amino acids has a very low B factor, they are surely going to form a partial helix-like structure. That's why it has assigned this partial helix structure. Now can you connect everything? Why there is a helix in between this loop and all?
The helix?
Now can you connect it? Why this particular thing has a helix in between, but not this two regions? Because these amino acids, as I've showed you, they have a very low B factor. So the molecule and the modeler or the ROBETA thinks that since this particular thing has a very low B factor, even in the loop, they must form a semi-helix structure. And that's why they are constrained. They are not moving a lot.
Okay.
Understood? Okay. So that's all how we can modify and use this PyMol to our benefit, and simply move around and use this to visualize the protein and find out the contacts and all. Okay. So now you can use your PyMol to visualize and hover around a particular protein, take pictures and all. Okay. Now there is another option called just click on file and see there is an option called export image as. Now if you simply click on the PNG, it is going to save your this particular picture in a PNG format. For example, say I have saved it on desktop, So let me show what is picture. Yeah, this is the picture. See, this is the picture. Suppose you need to show this picture on your thesis or something, right? So you can get a picture like this. Just save this picture. But since I don't have a license, my license has actually yes, khatam. It has finished.
Okay.
It's ended. So it's showing no license file for evolution only. 0 days remaining. Okay.
Oh. Mhm.
Okay. Yeah. So that's it about the PyMol. Okay.
Okay.
So we're going to in the next class we're going to start with the another server, the last server or the best server for protein modeling, the ITESR. I want to learn how that particular thing works. Okay, and then we're going to shift to the genomics areas and all. Right?
Mhm.
Okay,
Yeah.
fine.
Okay. Bye.
Okay, bye. Yeah, sure. Thank you.
Thank you.
Yeah, sure.